Alright, today we're looking at cyclin is degraded by the ubiquitin pathway. This was done by Michael Glotzer, Andrew Murray, and Mark Kirshner. This was published in 1991. So the objective of their experiment is they wanted to show that cyclin is degraded by the ubiquitin pathway, and they also wanted to ended up further characterizing like some like sequence stuff and everything like that. And they did some kinetics, like I taught the kinetics, but they did do a little bit of not like characterization of their kinetics. So some background information here is, first of all, is that proteolysis, which is the breakdown of proteins, is a major process in regulating the cell cycle, which we know this because cyclin, hence because cyclin has a name that its concentration fluctuates, so we already know that the breakdown of certain proteins throughout the cell cycle is a very important thing in progressing from one step to the next. Then they mentioned that mitosis is induced by something called M-phase promoting factor, or MPF. Then we know that, and they talk about how the transition from metaphase to anaphase, and really even more to interphase, is like the main signal for that is the degradation of cyclin. And then they also go and talk about how there's the mutants of cyclin that they can't be degraded. So essentially, mutants of cyclin that can still activate MPF, but they themselves can't be degraded. They pause cells in mitosis. So roughly around like the metaphase and anaphase checkpoint on there, they'll just get stuck because cyclin needs to be degraded, and if it can't be degraded, then they'll be stuck there. And they also obviously go on and say that polyubiquitination is the process of attaching a ubiquitin tail to a lysine residue, which marks a protein for degradation. And lastly, in this experiment, they studied xenopus, which we know is an African frog. So the methodology, again, for this experiment, just like the last one, their methods was a lot of it was very in depth, and I'm not going to get into it because I'll just be here all day explaining all the whole methods. But the main method that they had used is they had created a stable metaphase state to closely observe how cyclin is degraded, and cyclin is degraded constitutively. So essentially, what they did is they took some modified cyclin and they essentially injected it into xenopus cells in interphase, and then in the first figure I'm going to show is proof that this worked, that it creates a metaphase state in which cyclin is degraded constitutively. So here's the first figure that they use here. And this is showing that their method that I just described works, and so let's first break this down. So first of all, delta 90 at the top protein, it's a cyclin protein that has the first 90 amino acids missing. And so plus delta 90 means that protein's present, minus means it's not. The numbers on the side just correspond to masses, and cyclin delta 13 is also a cyclin that just corresponds to a cyclin that has its first 13 amino acids missing. And the numbers at the top represent times. And what they're trying to show here is that starting from 0 minutes all the way up to 40 minutes, that the cyclin concentration is going down. The bands are essentially slowly disappearing, and it's going down pretty rapidly. Or by 50 minutes, it's practically gone. Whereas then, right between 50 and 60, they add more of the cyclin 13, and then it goes back up, and then roughly by 80, it's gone again. And then they added more between 80 and 90, and then by 110, the cyclin concentration has gone again. And that's while the delta 90 protein is active. And while the comparison here to when their cyclin 90 protein is not there is showing that it takes till roughly 110 minutes after it's been added for it to really kind of disappear. So that first thing is just really trying to show that, hey, this actually worked, and this shows how they were actually able to test their theory and prove how cyclin's broken down. And B is showing it's a measure of histone 3 kinase activity, which is essentially just also trying to measure the activity of MPF that I had mentioned earlier, which is showing that mitosis is actually happening, which is also more proof essentially that CYC, or the cyclin, is getting broken down, which as you can see, the bands are bigger when it's being degraded, which I mentioned earlier is very important that it's broken down. So now let's get into some of their main results. So they had 2 sections of main results. Again, they did do kinetics, but I'm not going to go into it because that is not really that important to understanding the experiment, so I'm not going to talk about the kinetics. So the first thing is they talk about the sequences. So the first thing they wanted to know is they essentially what they did is they compared the N-terminal of the first 90 amino acids of 13 different cyclins across 8 different species. What they had discovered is they had only found 10 to 20% sequence conservation, which was very interesting. And the reason why that they did this is because they wanted to know essentially is, does the M-terminal by itself provide enough information for the cyclin to be degraded? And so, but anyway, so through this comparison, there was only 2 common sequences really. One RAALGNISN, which is just single letter abbreviations for amino acids. There are some variations in that. I'm not listing here, but there are some variations in there. But that is what they called the destruction box. And then the other thing I noticed is from amino acids 54 to 90, even though there's not a common sort of like a sequence of amino acids, they notice that there's a higher enrichment of lysine residues, which ranges from 1 to 13, which does make sense because that would seem to be where the polyubiquitination would happen. And then using their cyclin 13, so now they did a bunch of other sort of experiments here. So they had used cyclin 13, which already was missing the first 13 amino acids, and then they went on to delete residues 66 to 98, which left them essentially with their desired sequences that I had just mentioned. There were also some bonding motifs, some very short ones I didn't mention that were also present. And what they had saw is that this was still able to successfully be degraded in metaphase, but it was stable in interphase, which makes sense because in interphase, as the cyclin concentration is going to be going up, obviously because cyclins are needed for later on, so that would make sense. And then they did a larger deletion of 5498, and the different thing about this is it had produced cyclin that was stable in both states, which essentially means that this had arrested the state again in mitosis, because if it can't break down cyclin, it gets stuck in mitosis and can't move on to the next interphase. And then by performing similar tests on E. coli of deleting different sizes of residues and everything like that, they had determined that the amino acids 54 to 66 it must play a major role in cyclin degradation, because obviously they had seen similar things that I had just shown here. And it was like, wait a second, why is this, why is it not, it's essentially resulting in being stable or being degraded, or the degradation being blocked entirely. So through those things, they had determined that those amino acid sequences, which is also the destruction box, must play a very important role in the degradation of cyclin. And also, I didn't put it here on my slide, but is they actually, wait, I think it's on my next slide, I think. Okay, never mind. So, and now I don't have any figures for that because I didn't think the figures were too useful at explaining that. So, now we're on to the ubiquitination results. So, they had noticed, I do have figures for these though. So, just before the onset of cyclin destruction, they had noticed that there was what they called high MR bands on their gels. They were very faint, but they were present, which was a very weird thing because they didn't understand why it was there. They was very faint. And what they noticed was as these bands had also disappeared along with the degradation of the unmodified cyclin. And so they had produced a gel using 1391 PRA, which is a fusion protein, so PRA is a protein from sea urchins, and 1391 is amino acids 13 to 91 from the xenopus, I believe. And essentially, it came from, so the bands came from this because what they had done is they had iodinated this 1391 PRA fusion protein, and essentially what they had is just these high MR bands. So that's how because of that, they knew it had to come from these bands had to come from that. And that's big, as I kind of just mentioned here, is because that was the only protein that had labeled in these gels. So that's how they knew that had to be where the MR bands were coming from. Because they also had thought that these might have been ubiquitin 1391 PRA conjugates. So what they ended up doing is they also had they iodinated ubiquitin to the cell's interphase with and without the protein present. So that way they could or I should say with or without the protein labeled, not present. So that way they could ultimately figure out, okay, is the ubiquitin actually attached here or not? And they had produced bands they had produced bands regardless if it was incubated with the 1391 PRA or not, but I should say again it was labeled again. So these are the two figures they have here. So the ones on the left, the gel for A, so the gel labeled A on the left Essentially, if you can see, there's the faint MR bands that they were mentioning earlier, and which were the ones that were like, are these maybe the conjugations of the protein and the ubiquitin? And then B was a better representation of that, so you can clearly see that the bands are there, and they know those bands have to be coming from the protein because that was the only one they had labeled. And then the gel on the right shows the ubiquitin iodinized, and the main thing to notice here is that if you look at the ubiquitin, as you look at the ubiquitin M and iodine abstracts, the pluses, you compare those, because those bands are present with the iodine, with the 1391 PRA, but it's not actually labeled. And then lanes 5 and 6 also show so 5 and 6 have 1391 PRA that are labeled, while the ubiquitin is not labeled. And so the important thing here is to see that the bands are relatively in the exact same spot, which is more so evidence that they're proving that the high MR bands that they saw before must be conjugated pair, not conjugated pairs, but no, conjugations of the 1391 protein A bonded to the ubiquitin. And so essentially the whole main takeaway from all of this is that they had successfully shown that cyclin is broken down to advance from mitosis to interphase. Its degradation is very important for this to happen. And that cyclin is broken down by polyubiquitination. And essentially we really mainly have understand this today because we know that cyclin binds to CDKs. We know that CDKs are not actually active during mitosis. So if you have cyclin present, odds are CDK would be present, which means that it'll be phosphorylating things that didn't need to be phosphorylated, which would ultimately prevent this from happening. So, which is why cyclin needs to be broken down.